So now let's return to substituted benzoic acids and try and determine what's going to happen with different substituents. So first let's think about if we have an electron withdrawing group somewhere on the ring. We'll pull electron density this way, which will stabilize the carboxylate, which will make it the conjugate base of a better acid. Now, if we put an electron donating group onto our benzoic acid, that's going to push electron density into the ring, which will push more electron density towards our carboxylate anion, which will destabilize the carboxylate, which will make this a weaker acid. Another common category of organic acids are the sulphonic acids. One of the most commonly used ones is para-toluene sulphonic acid. Which is often called tossic acid for short, and is sometimes just written TSOH. Now, you should be well enough used to drawing resonance structures and determining pKa's that you should have a fairly good idea of what kind of an acid this is, whether it's a weak acid, strong acid, moderate acid, by looking at the structure. So take a minute to figure out what kind of acid you think this is, and then continue, and I'll talk about why it is the kind of acid that it is. So the pKa of para-toluene sulphonic acid is about -7. It's an extremely strong acid. Now, if you look at the structure here, with one OH and 2 double bonds between sulfur and oxygen, it's very similar in structure to sulfuric acid. And of course, the reason that sulfuric acid is such a strong acid is because of all the resonance stabilization that's available when you make the anion. Try to take para-toluene sulphonic acid and see if you can draw the anion of that, so deprotonate it, and then draw all the resonance forms of it. Pause the video until you finish that, and then we will continue. Alright, so you can see the negative charge is evenly shared among 3 different oxygens. The negative charge is very delocalized in this case, making this conjugate base very stable, which makes that proton very easy to leave. And I'm sure for a lot of you, one of the most interesting kind of acids that we're going to run into are the amino acids. Now, typically when we're talking about amino acids, we actually are only talking about alpha amino acids. There are all kinds of amino acids that can exist, but the alpha amino acids are the ones that are interesting in biology. With a general structure, so my amine is on this carbon here, which is alpha to the carboxylic acid. That's why they're alpha amino acid. Now there are 20 common alpha amino acids. And for our class, you don't have to memorize all of them, but I think there's some that are very interesting. And as we talk about those, there are 10 that can be synthesized by your body. And there are another 10 that cannot be. And these are the ones that we call the essential amino acids. For your diet, because you have to ingest these somehow. Your body can't make them. So you need to get these from your diet. So I'll show you a few of the amino acids just to get an idea of the different types of structures that are available. There are a lot of different R groups that can exist. R can be as simple as hydrogen, and that would be glycine. And glycine is interesting because it's the only of the alpha amino acids that has no stereocenter, because there are 2 hydrogens on the alpha carbon. You can get slightly more complicated where R equals methyl, and that's called alanine. Now I have to tell you that I'm kind of lying to you right now, and you'll see in a few minutes why that is, because the structures that we're drawing are not actually real structures. Okay, so that would be alanine, but this is a CH3 group, or methyl group. You can have aromatic groups on there. This is phenylalanine with a CH2 and benzene group on there. And you may have heard of phenylalanine before because NutraSweet contains phenylalanine. And so there's warning labels on cans of diet soda and other things that have NutraSweet in them that it contains phenylalanine, because there is a particular condition called phenylketonuria where people cannot break down phenylalanine. And so that way, if you take in a whole lot of phenylalanine in your diet, your body can't break it down and it'll bioaccumulate, and it's not good for you. We could have alcohols on there. So I could just have CH2OH. That would be serine. I can have amines on there. So if I have 4 carbons in an amino group, that's lysine. I can have carboxylic acids on there, like glutamic acid. So there are a lot of different types of groups that can be the R group. The acid-base properties of amino acids are very interesting because all amino acids have both acids and bases on them. So my amine has a lone pair of electrons here that make this a good base, while my carboxylic acid has an acidic proton on here, which make it a good acid. So all amino acids are both going to be acidic and basic. So the question that we need to address is, what is the structure of any amino acid? And this is where you'll find out that I've been lying to you a little bit, because all the structures I've been drawing up to now don't exist. What we need to think about is, under different conditions, what will the structure be? So let's look at very low pH. Okay, so under acidic conditions. So we'll take an example and let's do alanine. So the pKa of the carboxylic acid is 2.35, while the pKa of the protonated version of the amine would be 9.87. So that would be for the NH3+. So that means that at a pH of 1, below both pKa's, the structure is going to look like this. The carboxylic acid will have its proton, and the amine will be an NH3+. So the overall charge on the molecule is going to be +1 under acidic conditions. Okay, let's look at very basic conditions. At a pH of 12, this one will be deprotonated, and this one will also be deprotonated. We're above the pKa of each of those, so both of those will be deprotonated. So at a pH of 12, what will it look like? So at a pH of 12, carboxylate is now deprotonated, my ammonium is deprotonated, and my overall charge is going to be -1. That's going to be under basic conditions. So then what happens when we're between these two pHs? So at a pH of 7, for example. So at a pH of 7, we're above the pKa of this proton, so this one will be deprotonated, but we're below the pKa of this one, so this one is still protonated. So if I have to draw the structure at pH of 7, we have a carboxylate on this side, and we have our NH3+ on the other side. The overall charge here is 0, so this molecule is neutral and we're under neutral conditions. But you'll notice that there are charges. In every single structure that you draw of amino acids, there are always charges. So this structure that I've been drawing back here, this is what I was lying to you about. You can never have an amino acid that has the amino group neutral and the acid group neutral at the same time. Under acidic conditions, the amino group is protonated. Under basic conditions, the carboxylate is deprotonated. And under neutral conditions, the amino group is protonated and the carboxylate is deprotonated. So amino acids will always have charges on, no matter what pH they're at.